Hey, welcome. And on behalf of the Biochemical Society and Portland Press, I'm pleased to welcome you to today's webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via the text box, and we welcome suggestions for future topics and speakers to feature in our webinar series. And please see the website for more details on that. So my name's Nick Morton, and I am the Interim Honorary Meeting Secretary at the Biochemical Society, and I also sit on the Society's Awards Committee. In my day job, I'm Professor of Metabolic Resilience at Nottingham Trent University in the UK, so I'm particularly delighted to be able to chair this exciting awards webinar. Today we will hear from the Biochemical Society's 26th International Award winner, that's Dr. Lykke from the University of Copenhagen, and she's an Associate Professor in the Department of Biomedical Sciences. Just to note that this award recognises outstanding and independent research that illustrates the importance of the molecular biosciences in the advancement of life sciences research. So congratulations, Lykke. After completing a PhD in molecular physiology, Lykke started her own research group in 2021 focusing on molecular metabolism in cancer and aging. Lykke's work bridges fundamental biochemistry with translational applications, uncovering key biochemical mechanisms that regulate glucose uptake and muscle adaptation. And using innovative techniques such as adenovirus-associated virus delivery systems and inducible tissue-specific gene editing, Lykke has uncovered the interaction between muscle mass and insulin sensitivity via the TGF-beta pathway. And this has profound implications for metabolic disorders. Lykke's research has also shed light on the role of small GTPases such as RAC1 in glucose uptake, a pathway disrupted in type 2 diabetes and obesity. So before I hand over to Lykke to give her seminar, I just want to remind you that you can send questions during the talk by typing them into the questions box, which you should see here on the screen, and we'll try to answer as many as time allows. So I'm delighted to hand over now to Lykke. This looks good.
Very good. Thank you so much for this opportunity to present. It is a true honor for me to have been nominated and to receive this prize. And I really want to extend the honor to everyone that I've been working with over the years, the students in my team, PhDs, postdocs, my mentors, everyone whose shoulders the work we do stands on. And I'll do my best, of course, to take the opportunity that such a prize is to make the field of exercise biochemistry even stronger, because I think it's a very, very important topic. And in my team, we have focused very much on understanding the biochemistry of muscle metabolism and how exercise can influence this. And we know that every hour, every minute, and every second, there are all of these different biochemical signals that are turned on, they're turned off within the muscle, actually within all of our cells. And exercise very much influences these signals. And because exercise can really restore much of the defects that we see in many diseases, I'm very fascinated about how the signaling works in the skeletal muscle. And I actually see it sometimes when I stare up in the sky, look at the stars, that all of these stars are these different signals that are turned on and turned off. Sometimes there's a shooting star, it comes up, you can see it, it goes away. So I think this network of signals that are turned on and turned off are extremely fascinating. And because the effects of exercise are so powerful and so beneficial for our health, I think it's really fair to say that they are molecular magic. So I will try to show you some of what we have done in this field. And we know that exercise lowers mortality, and it does so probably by reducing the risk, but also reducing the risk of dying from many of the different diseases that affect us in the Western world. And that includes cardiovascular disease, type 2 diabetes, many types of cancer, also muscle wasting, sarcopenia, but also falls, depression, and dementia. All of these conditions, exercise has a beneficial effect. And I have been very focused on, with all our collaborators and the team that I work in, on trying to understand why it is that exercise is so beneficial for many of these different conditions, with a focus on the skeletal muscle and the biochemical signals that exercise elicits in the muscle that could then influence our health. And we have looked mostly in conditions of type 2 diabetes, but we're also getting a little more into the field of cancer, and especially cancer-associated metabolic dysfunction and muscle wasting, which I'll also show you a bit of data on. But before I go into this molecular magic of exercise, I want to show some of the key studies that really illustrate this powerful effect. And I will focus first on the most important endpoint, which I think probably is our risk of dying, which is something we're all very interested in delaying as much as we can. And one of the first studies that really showed the benefits of physical activity that I like very much is this one here, where from the 50s, where they have studied the risk of dying from cardiovascular disease in the drivers of these beautiful old buses in London, compared to the person who would go out, help the people come in, get the tickets. So these two people worked in the same environment. They were also more or less from the same social class, but one of the two had only half the risk of dying from cardiovascular disease compared to the other. And you probably have guessed that the person who had the lowest risk of dying from cardiovascular disease was this person coming up and greeting people, moving around, compared to the person who was just sitting, driving the bus all day. And after this study, many more, also more well controlled, but also bigger studies have shown the same effects. And here I'm just showing one more that I think is a famous study, because this is a study that sort of was the basis of why we are taught by our smartwatches that we should move 10,000 steps per day. But as you can see here, the largest improvement or the largest reduction in mortality rate actually comes from going from hardly anything to around 6,000 to 8,000 steps per day. So I have set my smartwatch to 8,000 steps per day, and then I get a little firework, which is a nice reward. But really, the reward, of course, is to lower the to lower the risk of dying. Another study that came out very recently was one that actually calculated the life expectancy gained in years from increasing physical activity. So here they looked at the association of moving more, so having more moderate to vigorous physical activity in minutes per day. And I think it was quite remarkable that just by going from 6 minutes to 15 minutes, this increases life expectancy by 2 years. So it's quite a lot gained from relatively little effort and time here. And of course, the larger benefits are given if you can move or be vigorously active for about 30 minutes per day, which is also the current recommended guidelines. So I think it's extremely relevant to understand the molecular mechanisms that mediate these effects. And in my lab, we have a very strong focus on the muscle and how the muscle helps improve our health. And I think that a better biochemical understanding of these processes is not only extremely interesting from a basic point of view, but it's also maybe something that one day can show us how to treat different diseases. So to understand this molecularly a bit better, an approach that has been taken by my lab has been to try and mine the different proteomics and phosphoproteomic studies that are coming out and keep coming out, and they keep becoming better and better with more and more information, trying to understand what is actually the molecular signature of exercise, acute exercise, but also what happens more over time if you exercise for a longer period of time. What is happening then in the muscle? And we've tried to look at some of these pathways, and a general take on it is that the minority of what happens during exercise are the regulation of proteins where we know what they're doing. We have a functional annotation to what they do. However, the majority of the proteins that are activated by exercise, we actually don't know exactly what they do. So this is what we're trying to look at in my team, because these omics tools are really fantastic for discovery, but they do lack the functional annotations of these players identified. So some of the mechanisms have already been identified by excellent researchers in the field. So we're looking at these a little more unexplored ones, and we do this by focusing on the 3 main benefits of exercise that we sort of think of in time. So if you think of doing one exercise bout, then you can think of what happens during the exercise bout, and one of the things that happens that's important for our health is that when we start exercising, there is a marked increase in the glucose uptake by the working muscle. And that is because the energy requirement in the skeletal muscle can nearly instantaneously increase by more than 100-fold during exercise. And therefore, glucose is needed to enter the cell, and the glucose needs to be oxidized. And how this happens in the cell is extremely important and something we have studied. Then after the exercise bout, there is actually still an effect of the exercise. So even if we have exercised, we go home, we sit on the couch and we watch Netflix. But then if we eat something, then the insulin that is needed to stimulate the glucose uptake into the muscles from what we eat, the sensitivity to this insulin is actually enhanced after an exercise bout. So this is pretty remarkable. So if I go exercise, lie on the couch, eat a candy bar, this glucose will enter my muscle cells faster because I have a sensitization towards insulin compared to if I didn't exercise before. And this can last up to 48 hours. So this is also remarkable, and this happens even after the exercise bout has stopped. And then the third phase that we think of is the phase that, or is what happens if you keep exercise training longer term. So if you have these repeated exercise bouts that last several weeks and months, then there are more long-term adaptations to exercise training that, for example, includes an increase in muscle strength, an increase in mitochondrial biogenesis that enables us to fatigue less, but also to become stronger. So all of these different phases are elicits molecular changes that improve our health. And we have these acute effects of a single exercise session, which is good news because then we can always go and have an exercise session, and that is never wasted because we always have these acute effects during the exercise and after the exercise. And then there is a more sort of long-term, but also longer-lasting adaptations to the exercise that are also interesting to understand how these are elicited molecularly. We can also look at them at what happens sort of molecularly at the transcriptome level, and here we can see that there are changes that happens acutely. The phosphoproteome also changes extremely fast, metabolome in the muscle as well. And then there are the more longer term, so the proteomic changes, the changes of strength, muscle mass, oxidative capacity that comes after longer term training. So when we're looking back on this iceberg, and we have tried to look at some of the less regulated pathways, a pathway that we have been very interested in is the pathway that is mediated by this Rho GTPase called RAC1. And RAC1 can be activated by exercise. I'll show you here in a minute. But the way we measure this is actually by measuring its downstream target, which is PAC. And PAC1 or 2, when bound to active RAC1, is then autophosphorylated and then is an active kinase to elicit the downstream effects. And in this very nice phosphoproteomic study that was done by Professor Lykke and Benjamin Parker and Christian Strini, we could see that the phosphorylation of PAC, this was then later Western blotted by Stefan Ravn in my group, that in response to endurance exercise training, RAC1 was active, and then in recovery back down in humans. And then the same people came in and had another 3 muscle biopsies taken by Erik Richter. But this time the subjects underwent sprint exercise. And here we also saw this increase in RAC1 activation. And we also saw this after resistance exercise training. So altogether, this really showed us that RAC1 was activated during exercise, but we didn't really quite know what role RAC1 had in response to exercise. But we did have a clue because before that, Amira Klipp had really pioneered some work, and I was very fortunate to get to spend some time during my master's thesis in Emma Kipp's lab in Toronto. And she had shown that this RAC1 PAC axis was necessary in order for glucose to be taken up by muscle cells. And we had later then built on this work, showing that PAC was phosphorylated in humans, and that this insulin-stimulated phosphorylation in human skeletal muscle, work I did when I was doing my PhD in Professor Richter's group, and that has later been followed up by Lisbeth Møller, a postdoc in the team, and with help of Kurt Højlund. We could see that this activation by insulin of RAC1 was decreased in obese and type 2 diabetic subjects. So we knew that RAC1 was important for GLUT4 translocation in response to insulin. We also knew that it was reduced in insulin-resistant states. So we had some idea, and we then wanted to look at the role of RAC1 in exercise-stimulated glucose uptake. And this is a very tightly controlled process. Under normal circumstances, GLUT4 cannot, or the GLUT4 vesicle needs to be inserted into the plasma membrane in order for the hydrophilic glucose molecule to be able to cross the hydrophilic membrane bilayer of the cell. And this is a very regulated process that, in response to muscle contraction, is stimulated by two, maybe more pathways, but two pathways that I've illustrated very simply here. One that is activated during the metabolic stress that occurs during muscle contraction, that then activates AMPK. And AMPK then is responsible for a downstream signaling cascade that releases the GLUT4 vesicles so that they can go to the membrane. But at the same time, this cross-bridge formations, this muscle contraction, also elicits a mechanical stress signal on the membrane that can then activate RAC1. And RAC1 is then also required in order for the GLUT4 vesicles to be translocated to the membrane and be inserted into to the muscle sarcolemma and T-tubuli. So we've looked a little bit at this mechanistically, we have done this by using a muscle-specific RAC1 knockout mice. So these mice, they lack muscle RAC1. And then we've tested, can they still increase their glucose uptake during exercise? And the answer to that is actually no, not fully. So they actually lack the ability to mobilize glucose into the cell during exercise. So that tells us that RAC1 is an important step for exercise-mediated glucose uptake. We knew already at that time that AMPK also played an important role for glucose uptake during and after exercise. But we didn't know if there was an interaction between RAC1 or AMPK. So this we set out to test, again using transgenic mice. And here we had mice that were wild type or that lacked AMPK, so they were kinase dead. So they had the AMPK, but the AMPK did not work. Or we had mice that lacked RAC1, or both. And then we incubated their muscles and stimulated them electrically to contract, and then we could use isotopic tracers to trace how much glucose is then taken up by these muscles. And as you see here, muscle contraction elicits an increase in glucose uptake, which is partially inhibited by either knocking down RAC1 or removing AMPK's kinase activity. So there's a partial reduction here, but then when we inhibited both pathways, there were additive effects. So that actually told us that these pathways were in fact activated in parallel, and that both were required to fully stimulate glucose uptake in response to exercise. So looking at this, we were still not sure how exactly it was that RAC1 was mediating a glucose uptake in response to a contraction. But we knew from non-muscle cell work that RAC1 is a very important subunit of the NOX2 complex. And the NOX2 complex is it's a lot of proteins that comes together in order for the cell to be able to produce reactive oxygen species. And if RAC1 is not there, this complex cannot assemble. So we wondered what happened to this in muscle. And And if we looked in human skeletal muscle, we could see that exercise did increase DCF fluorescence, indicating that ROS production were occurring during exercise in humans. And then together with Carlos and Thomas Jensen here in Copenhagen, we wanted to see what happened if we overexpressed active RAC1 in the muscle to the production of reactive oxygen species. And here we used a method using the component adeno-associated virus to introduce active RAC1, a method that I was fortunate to learn when I was in Paul Gregorovic's lab during my postdoc. And this overexpression of active RAC1 increased the expression of the catalytic subunits of the NOx complex. And it also quite markedly increased the amount of reactive oxygens that were produced during exercise in the muscle. So with that, we wanted to understand, okay, so what if we do the opposite experiment? What if we knock out RAC1 again? What then happens to this increase in ROS production during exercise? And what we found was that the exercise-induced production of reactive oxygen species were markedly lowered, and this is again Carlos and Thomas Jensen's work that really nicely illustrated this. So what role then did this play for exercise-induced glucose uptake? Is this a mechanism by which RAC1 mediates this signal? So we then next knocked out mutated one of the important subunits of the NOx2 complex, and then again placed mice on a treadmill, exercised them, having the muscles contract, and then injected isotopic tracers so we could trace where the glucose is going in these muscles. And we could see that in response to exercise, and similarly to the RAC1 knockout mice, there was a marked reduction in exercise-induced glucose uptake in the muscles that could not activate their NOx2 complex. So that really suggested to us that a mechanism by which RAC1 released the GLUT4 vesicles to go to the membrane was likely via a NOx2 dependent mechanism. But what is happening further downstream of NOx2 is something we still need to explore further, and we don't know exactly yet. So having outlined some of the pathways by which exercise can bring in glucose to the muscle, there's also this remarkable effect after exercise that I talked about, where you have this enhanced insulin sensitivity after the cessation of the exercise bout. And this is something that has really been pioneered by Professor Erik Richter, who was also my PhD supervisor and is still my mentor and friend, who noticed that after exercise, insulin's stimulatory effect on glucose uptake was enhanced. Here I'm just showing a more recent study by Professor Erik Richter, illustrating this during a meal test, where you see in the blue arrested legs glucose uptake in response to a meal. So it increases in response to a meal, but it does not increase as much as the prior exercised leg. So this really illustrates how much potential exercise has to then later improve the insulin sensitivity of the working muscle. And this had been known for a while, but Erik Richter really showed this, but it had been known because in type 1 diabetic patients, they had noted already a long time ago that if they took insulin after they had exercised, they would become hypoglycemic as compared to if they took the same exogenous insulin without having exercised before. But this was really explained by Professor Erik Richter, who is I think to many, and in particularly to me, a scientific superhero in this field. So really an amazing discovery, which is relevant to anyone who has insulin resistance. Another thing that happens in the hours after an exercise bout is that there is an increase in glycogen synthesis. And this has been shown quite a while ago by Bergström and Hultman. I think this was actually themselves being the experimental, the people in the experiment. But what they showed was that this is a non-exercised muscle, and this is an exercised muscle here, and then they show the glycogen levels of that muscle. And what you see in the black is that by exercise, there is a marked reduction in glycogen in the muscle because it's utilized for fuel together with the glucose that is brought in during exercise. But then after the exercise bout has stopped, the glycogen is resynthesized, and there is actually what is called a supercompensation, so an even higher degree of glycogen storage in the muscles that had been exercising. And some of the mechanisms for this has been outlined, but we have also tried to see if this RAC1 was involved in this process. So once again, we've used this muscle-specific inducible RAC1 knockout mouse, and we electrically stimulated one muscle to contract, leaving the other one rested, and then we waited 4 hours. And then we measured glycogen resynthesis. And what we saw was that in the control, wild-type control mice, there was an increase in glycogen resynthesis after the 4 hours of contraction. But this did not occur in the RAC1 knockout mouse. And this is probably because the RAC1 knockout mice, they also do not increase their glucose uptake during exercise as much. So this was also a new mechanism that we could show, and this was done by Stefan Rau in my group. All right, so I think so far the molecular magic of acute exercise is that firstly it stimulates muscle glucose uptake, and these molecular miracles or the molecular magic also increases muscle insulin sensitivity and promotes glycogen storage. And importantly, these mechanisms seems to actually also work in insulin resistant states. So that means that this is a powerful way to bring in the blood, or bring down the blood glucose by bringing in the glucose into the muscles that can very well handle and safely store the glucose. And this is important in many conditions that are associated with insulin resistance, such as type 2 diabetes, obesity, cardiovascular disease, inactivity also, and maybe also a little unknown is that insulin resistance is also a comorbidity of cancer that we have become a little more interested in. And here I think that exercise really has a potential to enhance glucose disposal, increase insulin sensitivity, and also improve prognosis. And I will show you why I think that. First, I just want to convince those of you who have not heard of insulin resistance in cancer that it is a thing. Here we have compiled all of the different studies that have used the gold standard, so the hyperinsulinemic euglycemic clamp, to study insulin sensitivity in humans. And they have then compared patients with various cancers to their respective controls within that study. And when we do this, we can see that the glucose disposal rate induced by insulin is much lower in the patients with cancer, and this is across the board. And the degree of insulin resistance in these patients is actually to the same degree as normally seen in patients with type 2 diabetes. So this is severe insulin resistance. And in line with that, we have also been able to see that there is an increased risk of developing type 2 diabetes if you get a cancer diagnosis, and that aligns with, well, we know that insulin resistance drives the development of type 2 diabetes. With regards to prognosis, we have also tried to look at the survival probability in patients with cancer who then got diabetes. And here you can see that the 70% survival probability for, and this is a plotted a prediction for a 65-year-old woman with a Carlson comorbidity index of 0 with or without diabetes. And you can see that if she predicted she could have her cancer diagnosis without diabetes, she would live about 2 years longer compared to if she got diabetes. So this is actually something that has real implications for patients with cancer, and where exercise could maybe really have a beneficial effect. And now I want to try and explain why there is this pretty incredible, I think, interaction between the muscle biochemistry, exercise, and the risk of cancer. Because I think this insulin resistance is very well illustrated in this graph, where you have glucose disposal, and then you have patients with colorectal cancer and controls. And in normal, healthy, lean individuals, insulin stimulates glucose uptake into the muscle in a dose-dependent manner, with a half maximal effect of around plasma insulin concentration of 60 microunits per ml. So that's what is needed in order to get the glucose in there. However, if you're insulin resistant, so cannot respond to the insulin, the body will still try to get the glucose in there because it needs to get in there. But in order to do so, a much higher dose of insulin is really needed. And this causes hyperinsulinemia, sometimes together with hypoglycemia. And this condition is something that is great news for tumorigenic cells, because they have a very high expression of insulin receptors, so they're very well equipped to sense insulin. At the same time, insulin promotes proliferation, survival, and metabolism of tumorigenic cells. On the other hand, then, we have exercise. Exercise sort of shifts this curve to the left. And that means in order to get the same glucose uptake, because of the increased insulin sensitivity, and also together with the enhanced glucose uptake into the muscle during the exercise bout that is independent of insulin, much lower insulin levels are required in order to maintain euglycemia. So exercise actually lowers insulin levels and probably also helps maintain glycemic excursions, which is not good news for tumorigenic cells. And this could and has been suggested as one of the mechanisms by which exercise potently lowers the risk of developing cancer, but also lowers the risk of death from cancer once one is unlucky enough to get cancer. And that is illustrated in this meta-analysis here. And the data is strong for this. And I think this interplay between the metabolism, the muscle, and the cancer is extremely interesting and something that I really want to understand a bit better. I also want to mention that the lowering of insulin levels up is probably not the only thing that helps prevent cancer by exercise, because exercise also improves the immune function and other things. All right, so having shown that the acute effects of exercise can really improve health and some of the molecular mechanisms that we've outlined, the next 5 minutes I will spend on looking at a little bit longer lasting effects of exercise training that also mediates the beneficial effects. And I'll focus on muscle mass and function here. So one of the things that happens with exercise training, we know for a long time, thanks to John Holossi's work, is that there's an increase in mitochondrial biogenesis and mitochondrial function that is illustrated here in human muscle, where you see sedentary and exercise training can very much increase mitochondrial respiration. And the mitochondria are really what produces energy for the muscle. So this is important for muscle function. At the same time, in the hours after exercise, there's an increased mTOR activity that builds myofibrillar proteins that is required in order to increase muscle fiber size and to build strength. So these processes are very important, and this is illustrated here in these young males, old males, where you see the cross-section of the thigh. We have this lowering of muscle mass that can be very much restored by exercise training thanks to the activation of these mechanisms. So this is important in cancer, in particular in cancer cachexia, which is a condition that occurs in up to 80% of patients with advanced cancer, and it has been shown to be the cause of death in 30% of these patients. So muscle mass is extremely important in cancer, and that is illustrated here as well, showing the overall survival in cachectic and non-cachectic patients. And here it shows that after about 4 years, only half of the patients with cachexia are alive, whereas it's about 80% of the patients without cachexia, so without muscle wasting, that is alive. So this is important. It has also been shown more in other populations, such as sarcopenia, where sarcopenia, so low muscle mass and strength, are associated with an increased hazard rate or odds ratio of different comorbidities, also increased risk of metabolic syndrome, increased risk of all-cause mortality, but also hospitalization stays are much longer if you have low muscle mass, which is particularly important for the elderly population. And if you're looking at the elderly population in particular, this study here I think illustrates very nicely how important muscle mass and function is. So if you're in the lower third of muscle strength, this is you have a very high mortality rate, whereas this is very much lowered in those third who have the higher muscle strength. And that is probably also why there is really a rush now from also medical companies to try and find drugs that can bump up muscle mass and muscle function. And targeted some of these pathways could really perhaps be important for understanding better what are the sort of molecular players that we need to target to increase muscle function. And I want to show you just one example for the last 2 minutes of one pathway we think is pretty exciting. And this is related to mitochondrial function. So we found one protein that was very much regulated by exercise in these omics studies. It was a protein called SLRP. As you can see here, it's heavily upregulated by exercise, but we had no idea what it was doing in the muscle when we discovered this some years ago. But we knew from non-muscle work that it was related to the mitochondrial OXPHOS complex assembly. Because we have the DNA, of course, that then encodes the mRNA, and many of the mitochondrial proteins are then imported into the mitochondria. But our mitochondria also has its own little tiny circular high copy number DNA. And that is also translated by the mitochondrial ribosome. And the stability of this mRNA is regulated by SLRP. So we thought, okay, what happens if we knock out SLRP? And this is work that was driven by Fung Pham, who's a postdoc in my group. And first she looked in humans. She could see that SLRP was increased with exercise training in men, but also in women. And when she knocked it out, we could see that the mitochondria here were extremely defragmented. We also did it in flies using the GAL4 system. And here you can see control flies and then SLRP knockout flies here. And then when we knock it down, the flies down, they will then automatically climb up. And what we noted was that these SLRP knockout flies, lacking SLRP only in the muscle of the flies, would climb up very slowly compared to the controls. So they had much lower muscle function, probably due to this decline in mitochondrial morphology or mitochondrial function. However, when we then placed them on treadmills, let the mice run. Mice like to run, which is a little unlike humans. So we got these exercise training effects after some weeks. These mice mitochondria were completely restored. So I think this is really a nice example of this molecular magic of exercise that can bypass even genetically induced abnormalities in, for example, the mitochondrial network. So with that, I hope to have somewhat convinced you that there is some molecular magic occurring in the muscle with exercise. I just want 3 take-home messages of how this transforms health. One is that it stimulates muscle glucose uptake acutely and independently of insulin, making it an extremely important therapeutic in insulin-resistant conditions, but also that it actually enhances insulin sensitivity and glycogen storage in the hours after the exercise. And finally, that it really helps build muscle mass and strength, which I think is extremely important, especially in an elderly population where sarcopenia and also hospitalization is quite common. So with that, I want to thank, I really want to sincerely thank my incredible team, but also our collaborators with whom I want to, to whom I want to dedicate and share this honor of this award. I also want to acknowledge all of the world in this field that I have not included here, because Here I try to focus mainly on our tiny contributions to the field. But so many amazing scientists and biochemists have really contributed very much as well. And of course, we're grateful for our funding and all of our collaborators. And with that, I also want to thank you very much for your attention. And I'll be very happy, of course, to take any questions that there might be. So thank you.
Okay, thank you so much, Lykke, for that fantastic seminar, a real tour de force in impactful molecular metabolism. And congratulations once more on your award. That's the International Award from the Biochemical Society. So we have time now for some questions, and I have many, no surprises, but I'm going to start with those that have been submitted through the text box. And the first question is from Aisha Coben, who asks, is RAC1 activation primarily an acute response to contraction, or does repeated exercise training induce long-term changes in RAC1 expression or sensitivity?
Yeah, so we mainly see that the increase in RAC1 activation is acute by one acute exercise bout. But we have seen evidence that in humans as well as in mouse muscle, that the total amount of RAC1 protein is also increased a little bit with exercise training. Whether the exercise-induced activation of RAC1 is also then increased after exercise training, we actually have not checked that. But the protein content can be increased.
Thank you. And we'll take a question from Katarina Kunzel, who asks, does it matter for the cancer patients which kind of exercise is done, endurance or strength training?
Yeah, I think this is a topic that has not been thoroughly studied enough in humans to test whether there are different exercise modalities that would be better for some patients than others. I think this is something we don't know yet. But with the knowledge of that some cancers are associated with metabolic dysfunctions, that is, for example, breast cancer and all other obesity-related cancers, here the focus should probably be, and I'm guessing because it has not been tested, but it should probably be to improve metabolic control. Whereas in other cancers, such as pancreatic cancer, lung cancer, where cancer cachexia is extremely common, I would believe, based on the current knowledge, that strength, like resistance exercise training, building up muscle mass and strength, would be more beneficial for those patients. But it is something that remains to be actually tested in humans. Yeah, but it's a good question.
Okay, and we have a question from John Bagter Jorgensen, I think. Can you elaborate on which muscle tissue depends on insulin receptors and insulin for glucose uptake, and which muscle tissue is only exercise dependent for glucose uptake?
So can you repeat that?
Yeah, I think the question, if I can just summarize, are there different muscle depots that are more or less driven, the glucose uptake is driven by the insulin receptor expression versus exercise?
Yeah, so generally, so if you look at the muscles within the body, the fiber type composition is quite heterogeneous. So there'll be type 1 fibers and type 2 fibers, and usually the type 2 fibers are a little less insulin sensitivity compared to the type 1 fibers, but they're both exercise responsive in humans. In mouse, it might be a little different, and I think there are different some groups showing that exercise enhances insulin sensitivity more in the type 1 fibers. But in humans, as far as I understand, it's exercise elicits the same effects on insulin sensitivity in both fiber types. And then we know that type 1 fibers also have higher content of mitochondria, which leaves them better at utilizing fatty acids, so fatty acid oxidation for exercise, for example. But mainly it depends on intensity, which fuel is used, probably more than the type of muscle. Yeah.
Very, very interesting. I had a similar question about the translation of the flies. You know, what do they have different fiber types? But I'll move on to maybe we can come back to that if we have time. I'll move on to a question from Grace Hepburn, who says, it was really interesting to hear how exercise influences insulin sensitivity. And I was wondering if there has been any investigation into other hormones such as glucagon.
Glucagon. So if exercise would increase the glucagon sensitivity. I actually don't know if there has. There probably has, and then I just may not know of them. Yeah, so I have to be. I have to owe you an answer to that one, I think. Yeah.
Great. Okay, so you can continue to put your questions into the box there. There are no more currently, but if that gives me a chance to ask a question, Lykke, about the, I wouldn't call it a dichotomy because I think you know the answer or might have a strong opinion. There is some evidence that calorie restriction is also protective against cancer progression. Yeah. So where do you find the balance, thinking about lifestyle and living with cancer, between exercise and calorie restriction?
Yes, it's a good question that I cannot answer in full at all. But if you look sort of at populations wise, there are populations that live under caloric restriction, and they do tend to have lower cancer incident rates. People who exercise also seem to have lower risk of developing cancers. And there might be an interaction. I think the risk of caloric restriction in cancer is that you also lose muscle mass.
Sure.
Which you do not do when you exercise. So with exercise, you still have the energy deficit signals, but without also reducing protein synthesis of the muscle. Whereas with caloric restriction, you have the energy deficit still, and those signals that can be beneficial because they can lower insulin levels, for example, lower glucose levels. But you also have the lowering of protein synthesis rate in the muscle, which is not beneficial, especially not for those cancers associated with cancer cachexia.
That's great. Okay, so we've got some more questions. So we have a question, I think Ellis Badrinwa managed to get in there first. So thank you, Dr. Lykke, interesting talk. Do you think exercise-induced sensitivity would also be beneficial in non-solid cancers? Apologies if covered in the talk and connection was lost there, so you might have to go over that again.
Yeah, I don't know with relation to the cancer as such, but in relation to the comorbidities of many cancers and many treatments as well, then exercise would be expected to benefit just general metabolic control, and therefore maybe reduce the risk of some of the comorbidities that are associated with cancer and anti-cancer treatment. But whether there are different effects of exercise in solid cancers compared to other, I actually don't know if there has been this stratification made as such.
Okay.
Yeah.
Okay, thank you. And we have a question from Abidi Mo. Building on the question about which type of exercise is best for cancer patients, how is this relevant to type 2 diabetes patients, and which type may be the best here based on current understanding?
For type 2 diabetes, the main goal should be to increase insulin sensitivity and lower the requirement, I think, as well for insulin, as well as reducing body weight. In cancer, the reduction of body weight is not always warranted because cancer often occurs in the absence of obesity, whereas type 2 diabetes is often in the presence of obesity. And here, exercise together with reducing caloric intake can help maintain a healthy weight loss. So I think the insulin sensitizing effect of exercise is extremely important in type 2 diabetes, but also there's been a lot of studies looking at the timing of the exercise in relation to the meal. So the idea has been that if you eat a meal and you have type 2 diabetes, then you can minimize the reliance on insulin to bring in the glucose by instead going exercising and then bring in the glucose from the meal in an insulin independent manner. And whether these mechanisms are similar or can be works in a similar manner and has similar benefits in cancer, I don't know of any studies that have looked at this yet.
Okay, well, we have another question coming in from Jessica Daniela, so I can't see the end of your name there, apologies. With so many pathways activated during exercise, how do you determine which ones are actually driving adaptation compared to just correlated responses?
Yeah, this is a big headache of studying this, I think, biochemistry, especially when we do it in vivo and in the complex contexts of diseases. The best we can do is to try to knock many of them out at the same time or inhibit several of them, because that can tell us something about which pathways are redundant and which are non-redundant. But I think it's in essence an impossible task. And with regards to the exercise induced, for example, bringing in the glucose in response to exercise, even if we completely block the ability to activate AMPK and RAC1 at the same time, we actually still saw an increase in exercise and contraction stimulated glucose uptake. So there's also something else which we don't know what that is yet. So it's really difficult to determine, and it also makes a lot of sense that these extremely important pathways have a lot of redundancy in them, because if we cannot bring in the glucose sort of evolutionarily, we would not be able to run away from danger or to find food in case one of them sets out. So it's an extremely difficult thing to study, I think. I don't have a good solution to that.
Great. Well, thanks for the answer. And just to confirm, it's Jessica Daniela Grania Alonso. I've expanded my text box to get the full names from some long names there. Thanks very much for that question, Jessica. So maybe I could ask another question, Lykke. You mentioned, you just touched briefly upon the immune system with exercise as being regulated. And I remember a lot of work done in exercise coming out of Denmark and Copenhagen on interleukin-6 and other cytokines, stroke myokines. So is exercise through RAC1, etc., also regulating the endocrine impact on cancer cells or even neurodegeneration in diabetes? Are there other factors beyond insulin itself that are at play here?
Yeah, we have never looked at, so this is a Bente Klarlund Pedersen, Mark Fabrio kind of work that has shown that IL-6 can be secreted during exercise, but also that there are other myokines that can be secreted and that can then sort of influence tumor growth. But also there are strong groups showing that the immune cell infiltration of the tumor is very much affected by exercise. Whether there's any relation to RAC1, we have no idea. We have never looked. I think RAC1 is probably mainly playing a part in the glucose uptake, metabolic part of things. We have never seen any alterations in immune cell, circulating immune cells or anything like that in RAC1 knockout mice, but we also have not looked specifically for that. And we know that there is a strong relationship between metabolic dysfunction and the immune system's ability to be activated when needed. And we know then that the RAC1 is important for these metabolic dysfunctions via its effect on GLUT4 translocation in the muscle. So maybe indirectly there might be some link, but I don't think there is a direct link.
Great, thanks very much. Final one for me. So I was looking at that survival curve with the increasing activity that you do, and you're making a very nice point that 30 minutes a day is the recommendation, and that buys you back some time in your lifespan or health span. Is there an aspect of that, looking at the far end of that curve, where you have to tell endurance athletes to pack it in and do less exercise? So are we, is there an indication that you might be going too far with exercise sometimes?
I'm sure that there is an extreme, and you also see, if you look at very, very extreme athletes, however, the number then of participants that can be studied are quite low. So I think it also needs to be taken with a pinch of salt, that we don't know that much yet, but there is a tendency towards a curve sort of leveling off, so that the benefits seem to first level off, but then maybe go back down. So there is some indications suggesting that there can be too much, but it needs to be quite a lot too much, and the sample size is low. So yeah, I think it's not conclusive.
Yeah, I know. Thanks for that. I know that's quite a controversial question because you don't want to discourage any form of physical activity for most people. Okay, so I think we've come to the end of our questions. So thank you very much, everyone, for attending, and for Lykke, our international award winner, for giving a fantastic seminar once more. You can continue that conversation online if you want to follow the Biochemical Society on Blue Sky, Instagram, or LinkedIn. So I just want to remind you as well that if you have any ideas for a biochemistry focused webinar for 2026, you can get more information about that at the website as well. All of those upcoming webinars are actually listed on the website, so if you've missed any, you can check in there to see for more exciting webinars, and you can visit their website or the YouTube channel. So finally, I think it's just great to highlight that it's more important than ever to stay connected and engage with our fellow molecular bioscientists, and particularly reaching into Europe. We're very privileged to have Lykke here speaking from Copenhagen. We've got tailored memberships that offer things to people at all career stages. So if you join the Biochemical Society's community of researchers and specialists, you'll be able to engage in cutting edge science and getting the most up-to-date information. So, of course, visit the website once again for more details on that. So I just wanted to thank everyone once again, and Lykke in particular, for a fantastic webinar. And thank you, everyone, and goodbye.
Thank you. Bye.